Hello everyone. Welcome to MedLive series of live webinars. I am Dr. Priyanka Sachdev and today I am going to talk about a small topic from general pathology. Just give me a second. So I am going to talk about a small topic from general pathology, that is cell death. We know that cell death are of two types, apoptosis and necrosis. In this lecture, I will be covering apoptosis. In the next lecture, I will be covering necrosis. So before starting apoptosis, I would like to briefly give an introduction. We know that human body is made up of cells. All the organs in our body are made up of cells. Normally, all the cells in the body are in a state of homeostasis. What do you mean by homeostasis? Homeostasis is a steady state. All the cells perform their functions within a narrow range, which is known as homeostasis. Just suppose this is a cell of human body. This can be any cell of the human body. It is performing its own function. For example, if it is a cell of stomach or GIT, it is doing absorption. If it is a cell of salivary gland, it is doing secretion. Whatever function is assigned to this cell, it is performing that narrow function, which is known as state of homeostasis. So normally, all the cells in human body are in a state of homeostasis, correct? But whenever there is stress to the cell. Now, what do you mean by stress? Stress is any abnormal environment around the cell, which is not normal, which is not usual. The stress can be of two types. It can be physiological, it can be pathological. Whenever there is stress to the body, whether physiological or pathological, cell do some steps. The first step, which after coming whenever a stress come to the cell, the first thing the cell do is adaptation. There are 5 types of adaptation we have already discussed in last lecture. What are the 5 types of adaptations? Hypertrophy, hyperplasia, atrophy, dysplasia, metaplasia. The 5 types of adaptation. These are always reversible. Whenever the stress is removed, the stress is withdrawn, the cell will remove the adaptation and come in its normal structure. That is known as cellular adaptation. Whenever a stress comes to the cell, the homeostasis stage is gone and cell will undergo adaptations. One of the or multiple types of adaptation, 5 types of adaptation. If the stress continues for a long time, or if the intensity of stress is too high, the second step which cell undergo is cell injury. Cell injury initially is always reversible. Whenever the stress is removed, the cell will take its normal structure and normal functions. That is reversible cell injury. But after a certain point, we can say point of no return, the cell undergoes irreversible cell injury. Irreversible cell injury, also known as cell death. Cell death is of 2 types, apoptosis and necrosis. So today we are going to discuss apoptosis. So can I say apoptosis is a type of cell death? Yes or no? Or can I say apoptosis is a type of irreversible cell injury? Irreversible cell injury are also known as cell death. So there are 2 mechanisms of cell death, that is apoptosis and necrosis. So is there any difference between the 2 mechanisms of cell death, that is apoptosis and necrosis? Definitely yes. How does a cell decide whether the cell wants to die with the help of apoptosis or necrosis? Is it a cell's choice? So what is the basic difference between 2 mechanisms? We will come on that point also, but later on. So in this diagram also, I have shown the same thing. That is, this is a cell. Normally, all the cells are in homeostasis. But whenever a stress comes on the cell, the stress can be physiological, can be pathological. If the stress is less, intensity is less or duration is less. If the stress is less, the first thing the cell undergoes is adaptation. The 5 types of adaptation, never forget, which are always reversible. Hypertrophy, hyperplasia, atrophy, metaplasia, dysplasia. I have already discussed all these 5, correct? But if the stress is more, either the intensity is more or the duration is more, the cell will undergo injury. The initial injury, the first injury is always reversible injury. But after a point of no return, if stress continues, high intensity stress continues for a long period, the cell will undergo irreversible cell injury. Irreversible cell injury is also known as cell death. The second name of irreversible cell injury is cell death. Cell death are of two types. We have already discussed apoptosis and necrosis. So I will start apoptosis. So what is apoptosis? Apoptosis, in this lecture, I will be discussing basically three things. What is apoptosis? That is, I will discuss the definition of apoptosis, which you can write in your university exams. Why apoptosis occurs? Why any cell wants to die? What is the cause? No one wants to die now. So why any cell wants to die? What is the cause of apoptosis? So we will discuss the causes of apoptosis. And the most important thing for university exams as well as for MCQs for your exams. How does apoptosis occur? That is the mechanism of apoptosis. There are basically two mechanisms of apoptosis, extrinsic mechanism and intrinsic mechanism. I will be discussing both of them in this lecture. So first I will discuss the what is apoptosis. That is definition of apoptosis. In short, apoptosis can also be called as cell suicide. Apoptosis can also be called as cell suicide. So why does a cell do suicide? No one wants to do suicide now. Why does cell suicide? Basically, there are some cells in our body which have already performed their function and body doesn't require those cells anymore. So those cells undergo suicide with the help of this mechanism, that is apoptosis. Secondly, there are some cells in the body which undergo cells damage. If cell damage occurs to any cell, there are repair mechanism. P53 gene comes and it does cell repair. But if the damage is beyond repair, the cell cannot be repaired anymore. The cell has to undergo suicide with the help of apoptosis. So can I say apoptosis is a type of cell death? Of course, it is a type of cell death. I got it. It is tightly regulated intracellular program. Now just suppose this is a cell. This is a cell. This is the nucleus of the cell. This is the DNA inside the nucleus. If the DNA is damaged to such an extent that it can't be repaired, or if the DNA is normal, it is not damaged, but the cell already performed its assigned function in the body and it is not required in the body anymore, then what happens? This cell, in the cytoplasm of the cell, there are certain enzymes which are known as caspase. These enzymes are known as caspase. Normally, these enzymes are inactivated. These are in the form of procaspase. These are present in the cytoplasm. But whenever the cell decided to undergo suicide, these procaspase gets converted into caspase. And these caspase will degrade the DNA of the cells and all the proteins of the cell, and cell will die. In this mechanism, in this manner, cell will commit a suicide. So in this mechanism, the cell which is destined to die, which has decided to die, will activate enzymes. What is the name of that enzyme? Caspase inside the cytoplasm. And that caspase will degrade the DNA and proteins of the cell and the cell will die. So don't mug the definition, just understand what does it mean. So that is the definition of apoptosis. So can I say apoptosis is a way of elimination of unwanted cells from the body? The cells which have already performed their function, or the cells which are damaged beyond repair. So those unwanted cells are eliminated from the body with the help of a mechanism which is known as apoptosis. So just note down the definition of apoptosis. It is a type of cell death which is genetically regulated, in which the cell which is destined to die will activate some enzymes which are known as caspase inside the cytoplasm, and those caspase will degrade the cell's nucleus as well as cytoplasmic protein, and cell will die. So it is a way of elimination of unwanted cells from the body. Correct. But in this whole process, there is no leakage outside. There is no leakage outside, so there is no inflammation. This is very important difference from necrosis, which is another type of cell death. In necrosis, there is leakage, so there is inflammation. But in apoptosis, there is no leakage, so there is no inflammation. Mark it. Make 5-star on it. Correct. So apoptosis is a programmed cell death. It is genetically programmed and it is energy dependent. If a cell wants to die, if a cell wants to commit suicide, ATP has to be consumed by the cell. Death is also not free in this world, correct? So I have already discussed what is apoptosis. Now you can write the definition of apoptosis very well. Now what are the causes of apoptosis? I have already discussed. The causes are of two types, physiological and pathological. So what are the physiological causes of apoptosis? What are the pathological causes of apoptosis? Physiological causes, the most important is embryogenesis. During embryogenesis, whenever an embryo is inside the uterus of the mother, whenever embryogenesis is taking place, how does this hand form? Do you remember your embryology? How does this hand form? The 5 fingers, 4 fingers and 1 thumb, how does this form? Initially the hand was like a block in this manner. It was like a block, but the cells between these 2 fingers undergo apoptosis. They undergo cell death. That's how these 2 fingers get separated. So in this way, apoptosis occurs during embryogenesis in multiple organ formation, correct? In the body, some lymphocytes become self-reactive. They will recognize cells as foreign. Such lymphocytes will cause autoimmune diseases to the body. So such lymphocytes get eliminated from the body with the help of apoptosis. This is the second example of physiological causes. During pregnancy, uterus and breast of the female, the size of the uterus and breast increases with the help of hypertrophy and hyperplasia. But after delivery, the size further reduces and become normal. So how does the size reduced further? With the help of apoptosis. So involution of uterus and breast after delivery, correct? And the death of the cells which have already completed their function. For example, there is a RBC. The lifespan of RBC is 120 days. In 120 days, the RBC has performed its all function, the gas exchange and all. But after 120 days, the RBC will undergo apoptosis, correct? So these are the physiological causes. Now coming on the pathological causes. For example, in AIDS, CD4 T cells, we know CD4 T cells undergo apoptosis. That's why their number is reduced. In Alzheimer's disease, the cells of the CNS undergo apoptosis. In MI, the cells of the heart, myocardial cells undergo apoptosis. So these are some diseases. You can mug up the name of 3 diseases basically here: AIDS, Alzheimer's disease, and MI. In AIDS, CD4 T cells apoptosis. Alzheimer's disease, CNS cells apoptosis. And in MI, myocardial infarction, myocardial heart cells undergoes apoptosis, correct? So causes of apoptosis, there are 2 causes, physiological, pathological. Write the examples of physiological. The main example there is embryogenesis, and in pathological you can write the diseases. Now coming on the most important part of this lecture, how does apoptosis occurs? That is the mechanism of apoptosis, correct? It is very interesting portion. So there are 2 mechanisms of apoptosis, extrinsic and intrinsic. There are 2 mechanisms of apoptosis, extrinsic and intrinsic. In both the pathway, extrinsic and intrinsic, there are 2 phases, initiation phase and execution phase. Execution phase of both the pathway is same. So I will discuss that part later. First, I will discuss the initiation of extrinsic phase. Then I will discuss initiation phase of intrinsic pathway. Then I will discuss execution of both, which is common. Execution is common. Execution is not different. In extrinsic as well as intrinsic, the execution phase is common. So I have to discuss only 3 things. Initiation of both, that is extrinsic as well as intrinsic. Execution I will discuss as a common thing for both. So I am starting. It is very interesting. Just listen. Just give me 1 second. What is basically 2 mechanism means intrinsic pathway and extrinsic pathway. In intrinsic pathway, the signal for the cell suicide comes from inside. That's why it is known as intrinsic pathway. For example, this is a cell you can see here. This is the nucleus of the cell. This is the DNA of the cell. If the DNA of the cell is damaged to such an extent that it can't be repaired, so this nucleus will send the signal from inside to the cell that cell, you should die now, because the DNA is damaged, you can't survive, you do the suicide. Signal is coming from inside, that is nucleus, that's why it is known as intrinsic pathway. In extrinsic pathway, signal comes in the form of ligand. Outside, some ligand will come and it will bind with the cell surface, and it will give the signal to the cell that your function is over now. Now you should suicide. So signal is coming from outside in the form of ligand, not from inside nucleus. That's why that mechanism is known as extrinsic pathway. So basically there are two pathways. Intrinsic, the signal will come from inside, and extrinsic, the signal will come from outside. First, I will discuss extrinsic pathway. So I'm starting extrinsic pathway. In extrinsic pathway, there are two phases, initiation and execution. First, I will discuss initiation phase of extrinsic pathway. Then I will discuss initiation phase of intrinsic pathway. Then I will discuss execution of both of them. Correct? So extrinsic pathway, I will show you in this diagram. Please have a look. Please everyone be here. So this is a cell. On every cell of the body, there are some receptors. Just a second. There are some receptors. These receptors are known as fast receptors or CD95. I have marked with red on the cell surface. Can you see on the surface of the cell there are receptors? These receptors are known as fast receptor, FAS fast receptors, or CD95 receptors, or death receptors. These are also known as death receptors. These are present on every cell of the body. Correct? But whenever some ligand comes from outside, we are discussing extrinsic pathway. So ligand will come from outside. Can you see this yellow colored ligand? Can you see here this yellow colored thing? This is ligand, fast ligand. This is coming from outside, taking the signal that cell should die. When this fast ligand will bind with the receptor, what will happen? I will show you in the next diagram. The red color thing in this diagram is fast receptor. The yellow color thing is ligand. Ligand will bind with the receptor. Can you see? When ligand binds with the receptor, all the receptors which were present far apart on the cell, they comes closer. They comes closer and form a domain. So all the receptors come closer and form a domain on the inner side of the cell membrane. They form a domain. This is known as domain. It is known as FADD, fast associated death domain. FADD, it is known as fast associated death domain. So what is happening? Whenever a ligand comes, whenever it combines with the death receptor or fast receptor, multiple fast receptor which were present apart, they comes closer on the cell membrane, and on the inner side of the cell membrane they form a domain which is known as fast associated death domain. Due to the formation of this domain, procaspase 8 which is present in the cytoplasm in inactive form gets converted into caspase 8. It gets activated. Caspase 8. So procaspase 8 gets converted into caspase 8 due to the formation of this domain. So that was all about initiation phase of extrinsic pathway. What happens next after the activation of caspase 8, I will tell you later on in the execution phase. Correct? So this is the initiation phase. This is a diagram from Robbins and Cotran. Very good book. You can see in this diagram. So this is the fast receptor with the red color on the cell membrane. A fast receptor is present. This yellow color thing is fast ligand. Can you see fast ligand? Fast ligand is binding with the fast receptor. So multiple fast receptors come closer to each other. Can you see? Multiple fast receptors come closer to each other and they form a domain. This domain is known as FADD. What is the full form of FADD? Never forget, fast associated death domain. Due to formation of this domain, procaspase 8, which is present in the cytoplasm in inactive form, gets converted into active caspase 8. Correct? So this is the flow chart you can draw. Always draw flow chart in your university exams. Never write lengthy paragraphs. Always draw flow charts, neat and beautiful diagrams if you want to secure more marks. Correct? So here, this is the fast receptor CD95. I'm sorry, just a second. Whenever fast ligand comes and binds with it, multiple fast proteins come together. They will form a domain which is known as fast associated death domain, due to which procaspase 8 will convert into caspase 8. I will continue the story in execution phase. So this is initiation phase. Initiation phase of extrinsic pathway is already over. I have finished it. Now I am coming on initiation phase of intrinsic pathway. Execution pathway I will discuss in common. I have already told you. So I am coming on the initiation phase of intrinsic pathway. Here the signal will come from inside. That's why it is known as initiation pathway. Intrinsic pathway. In extrinsic pathway, the signal was coming from outside in the form of ligand. Here the signal will come from inside. So we will see. So again, this is a cell. Here the signal will come from the nucleus. Just suppose this is the nucleus of the cell. I will draw it for you. So this is a cell. Please be here. This is a cell. This is the nucleus of the cell. This is the DNA of the cell. Just suppose DNA is damaged to such an extent that it can't be repaired. So this nucleus will send a signal to the mitochondria. Now I will draw mitochondria. So here is the mitochondria. Here the key component of the cell is mitochondria. There are two membranes in the mitochondria, we know, inner membrane and outer membrane. So I have drawn the two membranes of the mitochondria. Between the two membranes of the mitochondria, there lies a protein which is known as cytochrome C. So here I am drawing a protein which is known as cytochrome C. Cytochrome C can't come into the cytoplasm. It is present between the two membranes of the mitochondria. There is a door in the outer membrane of the mitochondria, but this door is closed with the help of a guard. What is the name of that guard? The name of that guard, there are guards. What is the name of that guard? Bcl-2 and Bcl-X. These two guards keeps the gate closed. This gate is closed. So cytochrome C cannot come outside into the cytoplasm because the gate is closed. This gate is known as mitochondrial permeability, transit MTP. Mitochondrial transit permeability is closed. This door is closed with the help of guards. These two guards do not permit cytochrome C to go outside. But if this is a normal situation. In this situation, there is no DNA damage. But just suppose if DNA damage is occurred, so this nucleus will send a signal to the mitochondria that DNA is already damaged. Now cell should commit suicide. So what does the mitochondria do? Mitochondria will open the gate. But how does it open? Guards won't allow to open the gate. So mitochondria will replace these two guards with another three guards. What are those three guards? What is the name of those three guards? These three guards are BARC, BIM, and BAX. BARC, BARCs, and BIM. These three guards are there. If these three guards will come, they will open the gate. Once the gate is opened, this cytochrome C will come outside into the cytoplasm. If cytochrome C gate is opened, cytochrome C will come outside into the cytoplasm. Once the cytochrome C comes into the cytoplasm, what happens? In the cytoplasm, there is procaspase 9. There is a pair, procaspase 9. Procaspase 9 is inactive. In that mechanism, there was procaspase 8. In this mechanism, there is procaspase 9. It forms a pair with another molecule which is known as APOF1. It forms a pair with APOF1. APOF1 keeps it inactivated. This cytochrome C will separate APOF1 from procaspase 8. So procaspase 8 will get converted into caspase 8 and it gets activated. So this is the whole mechanism. So what does, I will show you in the diagram. This is a diagram from Robinson-Cotran. So you can see this is the inner membrane of the mitochondria. I'm drawing it for you. Sorry. This is the inner membrane of the mitochondria. This is the outer membrane of the mitochondria. Between the two membranes, there lies cytochrome C, but the door is closed. It can't come outside. The door is closed. Door is closed with the help of Bcl-2 and Bcl-X. But once nucleus sends signal to the mitochondria that cell should die, these two guards are replaced by three another guards. What is the name of these three guards? BARCs, BAC, and BIM. These three will open the door. Once the door is opened, cytochrome C comes outside. Once the cytochrome C comes outside, in the cytoplasm, there is a pair. What is that pair? Procaspase 9 with the green color and APOF1 with the purple color. This APOF1 keep procaspase 9 in active form. So cytochrome C separate APOF1 from procaspase 9. Once it is separated, pro-caspase 9 gets converted into active caspase 9. So that is the initiation phase of intrinsic pathway. Here the signal comes from the nucleus to the mitochondria that cell should die. In that pathway, the signal was coming from ligand. It was coming outside. Correct? So that was all about intrinsic phase of initiation pathway. This is a flowchart for you people. So whenever stimuli comes from the nucleus to the mitochondria, on the outer membrane of the mitochondria, two guards are there, Bcl-2 and Bcl-X. These are replaced by three another guards, Bax, Bax, and Bim. They will open the door, that is MPT, mitochondrial permeability transit. Once the door is opened, cytochrome C will come outside into the cytoplasm. Once it comes outside, it will form a pair with Apop1. It will separate Apop1 from procaspase 9, and procaspase 9 gets converted into caspase 9. Correct. So I have covered what I have covered. I have covered the initiation phase of both pathway, intrinsic as well as extrinsic. Now you people tell me, what was the end point here in extrinsic pathway? Initiation phase of extrinsic pathway. What was the end point here? Here the end point was activation of caspase 8. Here I left the story. I told you I will continue it later. And here the end point was activation of caspase 9. So there I left the story activation of caspase 8. Here I left the story at activation of caspase 9. In execution phase, I will continue the story from here. Execution phase is common for both. It is the convergence point. Whether it is intrinsic pathway or extrinsic pathway, execution phase is common for both. In extrinsic pathway, there was caspase 8. In intrinsic pathway, there was caspase 9. So their caspase 8 and their caspase 9, they will activate caspase 3 and 7. The next two caspase are caspase 3 and 7, never forget. So the first caspase to be activated in execution phase of any pathway is caspase 3 and 7. The first caspase in intrinsic phase of extrinsic pathway is caspase 8. The first caspase in initiation phase of intrinsic pathway is caspase 9. Correct. Once all the caspase get activated, the nucleus and cytoplasmic proteins of the cells get degenerated and cell dies. Finally, phagocytosis of the cells occurs. I hope you understand the mechanism. So in this lecture, we have discussed three things. Apoptosis, what is apoptosis, why apoptosis occurs, and how does it occurs. The mechanism of apoptosis is very important for your university exams as well as for NEET and AIMS and all other competitive exams, MCQs of those exams. So in the next lecture, I will be discussing necrosis. Soon I'm coming live to teach you in live webinars. If you like the lecture, let me know. And if you have any particular topics, tough or difficult topics you want me to be taught by me, so you can message me. So thanks a lot. Thank you. So this is my Telegram channel. You can join my channel and let me know the topics which you want. So that is MedLive by Dr. Priyanka. This is my Facebook page, MedLive by Dr. Priyanka. You can also join my website, access my website, and let me know the topics which you want to be taught by me. Thank you. Bye-bye. See you.